Good afternoon. I am Dr. Charu Dogra Rawat from Ramjas College, University of Delhi, and I have been taking a series on recombinant DNA technology. Continuing in this series, today we will talk about the construction and screening of DNA libraries. The learning objectives for today's session would be to understand the construction of genomic library, to understand the construction of cDNA library, and to understand the library screening procedures. Now, when we talk about recombinant DNA technology, primarily our idea is to clone a gene of interest into a vector for any downstream processes which are helpful for the biotechnological industry. It can be for the sequencing of that particular gene sequence, it can be for the production of protein of that gene sequence, or it can be for any other thing which is required in the biotechnology sector. When we want to construct only one gene of interest, that was what we studied about the construction of recombinant DNA molecules. Now, sometimes it is necessary for the entire genome of an individual to be represented in these cloning vectors. If you recall, the cloning vectors were basically the vehicles that were required to carry this gene of interest or the region of interest into a host organism where it multiplies or it expresses its protein in abundance or any other downstream processes that we want to do with the gene of interest or the region of interest. But when we talk about the entire genome representation in a vector, then it is termed as the genomic library. The references used for this session, particularly for educational purposes, is T.A. Brown, which is a gene cloning and DNA analysis and introduction. Then the Twyman book, which is Advanced Molecular Biology, a concise reference. So, genomic library, as I was talking about, is a set of recombinant clones that contain all of the DNA present in an individual organism. So, this is actually a collection of recombinant clones. So, what are recombinant clones? They are the vectors that carry our gene of interest. So, if we divide the entire genome into a set of fragments and those fragments we clone into vectors, then this set of cloning vectors containing the gene of insert, which is the recombinant clones, represent the entire genome of that organism, and it is termed as a genomic library. A genomic library is a collection of clones sufficient in number to be likely to contain every single gene present in a particular organism. Now, this is a very interesting statement because when I say that we clone the entire genome into different cloning vectors and that set represents a genomic library, it becomes essential that all the genes or the entire genome divided into the fragments is represented in that genomic library. So, for that particular organism, every single gene is carried in the recombinant clones that constitute the genomic library. So, this is important. So, how do we go about preparing this genomic library? Now, genomic libraries are prepared by purifying the total cell DNA. So, again, if you recall the simple recombinant DNA technology technique, then we used to have this gene of interest which was coming from some source, DNA from some source what we have, and then we clone it or we insert it into a cloning vector. We also talked about the various types of cloning vectors such as plasmids, phage-mids, lambda phage based vectors or M13 phage based vectors, P1 vectors. We talked about the bacterial artificial chromosomes, P1 artificial chromosomes, yeast artificial chromosomes, all different kinds of, even for plant we talked about the vectors. We talked about the vectors based upon the viruses such as adenoviral vectors, retroviral vectors. So, we have already covered all the different types of cloning vectors in our earlier sessions. So, when we want to clone and have the representation of the entire genomic library, then the first thing is that we purify the total cell DNA from an organism. We have also covered the methods for the purification of the total cell DNA as well as the plasmid DNA in our earlier sessions. So, when we purify this total cell DNA, then we make a partial restriction digest. Now we talked about the restriction digestion, in which we cleave the genomic DNA or vector DNA, plasmid DNA or any nucleic acid with nucleases that cut inside the nucleic acids, therefore they are termed as endonucleases, and they cut at specific sites. So they recognize certain sequences and that they cleave at certain sequences, and therefore they are termed as restriction endonucleases. So we use these restriction endonucleases to digest the DNA. So to digest the total cell DNA, if you notice, we have used a word which is called as a partial restriction digest. Now when we want the entire genome to be represented in clones, our purpose is, after the cloning experiment, after the genomic library, one of the technique can be that when we sequence them, then we try to reconstruct the entire genome. So basically we know the fragments, we know the sequence of the fragments, and then we somehow join them together so that we get the sequence of the entire genome. Now we can have or reconstruct the entire genome only if the sequence has certain overlaps. So having those overlaps, we do a partial restriction digestion, because one fragment can be represented in more than two of the fragments that can be inserted. So when we sequence these clones, we can find the overlaps, the starting or the end positions, and then can arrange them in a linear fashion. So this is important. There can be other methods also which do not require partial restriction digest, but generally, as we are talking about the representation of the entire genome, our idea is that we can also reconstruct the entire genome from this whole library, or at least we should know what comes before, what comes after. That is also one of the purposes that we do this experiments for, and for that we use the partial restriction digest. Now these partial restriction digest resulting in fragments that can be cloned into a suitable vector. So just like a recombinant technology, usually a lambda replacement vector, a cosmid, or possibly a yeast artificial chromosome, bacterial artificial chromosome, or P1 vector. Now why have I just put these vectors? Why am I not including just a simple plasmid here? Because when we have to clone the region of interest or clone the entire genome into vectors, we require the vectors which have higher capacities, which take up large DNA fragments. So if you go back to the sessions on cloning vectors, you will see that the plasmids can carry up to 10 kb of DNA, foreign DNA in itself. However, if we use higher capacity vectors such as cosmids, they carry around 35 to 45 kb of DNA, yeast artificial chromosomes which can carry around 600 kb of DNA, or bacterial artificial chromosomes which can carry around 300 kb of DNA. So larger the fragments, lesser number of clones would be required to represent the entire genome. So that is why it is these vectors which are mentioned here. So this is a scheme that shows the basic ideology or basic technique behind the cloning of the construction of the genomic library. So you have a DNA, you cut it with different restriction enzymes. For example, in here an example is given that it is cleaved by SAW3A. Now you can see that when SAW3A cleaves the total cell DNA, you get different fragments. Now because if it is a partial digest, you will get different sized fragments and there will be overlapping regions in those also. Even if it is a complete digest, you will still have different sized fragments because the distribution of restriction sites are not uniform in the entire genome. So you will get different sized fragments, and these sized fragments are cloned in the cosmid. So we have again talked about the cosmids, and therefore we know when we clone the inserts into the cosmids, then the cosmids carry these cos sites which are the cohesive sites, and therefore they form these kind of catenanes which are there. So where many multiple fragments are cloned with cos sites at both the ends. The cleavage occurs, they cut at the cos sites, finally they are packaged into the viral heads and they are used to infect or introduce into the E. coli vectors, and therefore so this particular plate along with many of the different plates will represent a genomic library. So if the entire genome cloned in these cosmid vectors are introduced into the host E. coli, for example, in this case, then when these colonies appear, then all of these colonies would contain a recombinant DNA. Obviously, we will do some kind of a selection here, but if they contain the recombinant clones containing the insert of the DNA, then this would represent a genomic library. Now again a question comes in that how many clones do we require so that we assume that the entire genome has been represented. So there has to be a certain way. For example, if a cloning vector is taking 17 kb of genome, then in 17 kb of genome fragments, then how much will be required? How many number of clones do we need? Obviously, larger, the number of clones, the representation would be more. But it would again depend upon the size of the DNA. It will depend upon the size of the fragments that you are being created. It will depend upon the capacity of the vector, how much of the DNA can it take, and that would define the number of clones that would be required so that you can be get an idea that the entire genome is represented in those recombinant clones. So to find out the number of clones needed for a genomic library, there is a formula, and that is equal to ln 1 minus p ln 1 minus a upon b, where n is the number of clones that are required. So we need to find out that after transformation of the recombinant clones, how many colonies should we look at, that okay, if we have got these many colonies, our genome is represented. So that is calculated by P is the probability that any given gene will be present. So obviously there will be a probability that any given gene will be present in that library. A is the average size of the DNA fragments inserted into the vector. So A would be the size of the DNA fragment. Obviously average, because as I said, there will be different size of DNA fragments that would arise, and B is the total size of the genome. So depending upon the total size of the genome that you want to clone and the size of the DNA fragments, and then what is the probability of every gene to appear in the number of clones that is required can be calculated. So generally, whenever a scientist is constructing a genomic library, it tries to put the values in this particular figure and look at that, okay, these many number of clones we require. So because it is dependent upon A, which is the average size of the DNA fragments, this reflects the use of the vectors. So what are the capacity of the vectors? If there are high capacity vectors, that this A would be larger and the number of clones that would be required would be lesser. So if we look at this table basically tells us about the genome size of different organisms, and then it also talks about the number of clones that is required if the size of the fragment is 17 kb or if the size of the fragment is, for example, 35 kb. So if we are cloning in cosmids, then it is 35 kb. So if you look at the human, then around 554,000 clones are required if you cut the entire genome into 17 kb size fragments and clone it into a vector. So these many number of clones would be required. If you get these number of clones, then you can probably assume that the entire genome has been represented. And for example, if you use 35 kb fragments, then again some 200,000 clones are required. So it directly reflects that higher capacity vectors if you use. So if you want to clone a particular genome, you must be aware of the Human Genome Project. So in Human Genome Project, what they did was shotgun sequencing. We will talk in detail about these shotgun sequencing and Human Genome Project in our later sessions. But right now, what I can just let you know is, because it is related to the library, that what they did was that they divided the entire genome into smaller fragments. Now they did partial digestion, because as I said, they need to construct the genome also. So they did partial digestion, they cloned it into recombinant clones, into vectors, cloning vectors, and then they introduced these recombinant clones into the host bacteria, and then they started sequencing. So when they started sequencing, then they found out the overlapping regions between the clones and tried to try to ascertain the genome sequence, and it took some like 10 years to do this. Nowadays genome sequencing is hardly a matter of like 10 to 15 days, but I'm talking about when the human genome project was first decoded. So they assumed that if we are getting this many number of clones, so they used different types of vectors, not they used different type of vectors, but they could have used, and therefore they could have understood that there are so many number of clones which are required. So bags, for example, they can handle DNA inserts up to 300 kb in size, reducing the size of the human genomic library to just 30,000 clones. So from 200,000 clones, you just require 30,000 clones. Even more than that, if you use YAC vectors, which is yeast artificial chromosomes, if you use YAC vectors, they are routinely used to clone 600 kb fragments. And special types are able to handle DNA up to 1400 kb in length. So you can well imagine how much your number of clones needed for the representation of the entire genome will reduce if you use high capacity vector. Now the latter bringing down the size of the human gene library down to just 6500 clones. So after transformation, if you get 6500 clones plus in the fragments cloned in YAC vector, you can assume that your entire human genome is represented, and probably you need to just analyze these 6500 clones. But why do we not use them, or why do, what are the limitations in that? The major limitations in these mega YACs is the insert stability. The cloned DNA sometimes becoming rearranged by what is called as intramolecular recombination. So we don't want to do that. If we want to understand the human genome, we want to clone it into vectors, but we do want to clone it as it is. We want to sequence as it is. We don't want it to undergo any kind of modifications, changes, intramolecular recombination, or we also do not want that if we have got 7,000 clones, which is required, more than required, but then there is the loss of insert that occurs. So insert instability is there. So we don't want that. So depending upon what our need is, how many clones do we need for representation, what do we want to do in the downstream purposes, we choose the cloning vectors and clone the entire genome into these vectors to represent a genomic DNA library. Now this genomic DNA library, as I said, represents the entire genome, but this entire genome might not be expressed. It might not be, we very well know about the differential gene expression. When we talk about different cell types, then each cell type contains the same complement of genes. However, they do not represent the same repertoire of proteins. A muscle cell, a brain cell, they all contain a skin cell, they contain all the same set of genes. But the proteins or the genes that are expressed, they are different in muscle, they are different in kidney cells, they are different in skin cells, they are different in lung cells, and this is termed as differential gene expression. So when we want to talk about the functionality, we basically do not talk about the DNA as such. We talk about the mRNA, or we basically talk about the proteins. We talk about, because all the functions that are manifested in a cell are mostly by proteins. There are some catalytic RNAs also. There are, siRNAs, there are regulatory RNAs which are also altering the functionality or defining the functionality of a cell, but most of them is governed by the proteins that are formed. So we are more interested in, or sometimes we are more interested in finding out the protein aspect. We are finding out the genes. We want to know the genes which are expressed. We don't just want to know the entire genome sequence. So in multicellular organisms, in different types of cells, the same complement of genes are present. However, not all genes are expressed, and this is termed as differential gene expression. Not all genes can be expressed at all the time also. That is termed as gene regulation, because the genes would be present depending upon the conditions, depending upon the stimuli. Some of the genes would be expressed in one stage, and after that the other genes would also be expressed. So not all genes are expressed, not all genes are expressed at the same time. So there is a differential gene expression as well as there is a gene regulation that is going on in the cell. So only those genes that are being expressed are transcribed into mRNA. So the starting material for a library can be mRNA. If we are interested to study about the genes which are actually functional in a particular cell type, We would like to, we would be more interested in having a library that represents the mRNA rather the DNA. The DNA for the sequencing or the genetic content, knowing about it is good. But if we want to know the functionality, or we want to know in different cell types what are the genes that are actually expressed, or at a different cell times what are the genes that are expressed, then DNA will not solve the purpose. Our starting material is a library formed of mRNA. But mRNA is not a stable molecule. It cannot itself be ligated into a cloning vector. Therefore, what do? We convert this mRNA or the messenger RNA into DNA by complementary DNA synthesis. Now, you know about this complementary DNA synthesis because what happens is, in this case, the DNA is constructed or generated using mRNA as a template. Many cells have this, and even viruses have this enzyme which is termed as a reverse transcriptase. So, transcriptase or transcription is the conversion of DNA into RNA, not conversion, decoding of the DNA into RNA. So, decoding of DNA into an mRNA is basically termed as transcription. And you know that there are enzymes, which is RNA polymerase, that catalyzes this process. Now, in case we want to convert the mRNA into DNA, then some of the cells have this enzyme which is called as reverse transcriptase. So, reverse transcriptase will reverse the transcription mechanism and it will form the DNA which is then termed as the complementary DNA from the messenger RNA. But important thing is that these cDNA is now a representation of the expressed complement of the genes. Not the entire set of genes, but only the expressed complement of the genes will be represented by these cDNA molecules. Resulting cDNA clones are representation of the mRNA which is present in the original preparation, which is actually a representative of the genes expressed in a cell. So, I hope this point is clear that sometimes we do not want the sequence of the entire genome. If we want to understand the arrangement of genes, the organization of genes, the genetic content which is present, we go for a genomic library. But if we want to understand about the functional aspects, that what genes are expressed, what is the, the transcriptome if we need to talk about. We want to talk about in later case the proteome, that what are the genes that are expressed, what forms the proteins, what functions it is carrying out, how different cell types, different cells are carried out. Then we are more interested in constructing a library of cDNA rather than the mRNA, rather than the DNA, genomic DNA. This construction of library of cDNA is also very important in case of higher eukaryotic cells, because you know the eukaryotic cells have split genes. The genes which contain exons, which are the coding sequences, as well as the intervening sequences, which are the introns. So, if we want to know the genome organization, if we want to know the genetic content, then we go for the cloning the entire sequence. But if we do not want to know that, we basically want to talk about the exons which are joined together, then this we talk about in terms of again mRNA. So, cDNA library comes into us with lot of help when we want to understand the eukaryotic system or the eukaryotic system machinery which is there. So, looking at the cDNA cloning for preparation or cDNA library, we will continue with that. Thank you.